G'day, I'm Dr. Philip Button from the Food Microbiology Academy in Melbourne. Now what I want to do in this short video is take you through a portion of my honours research from 1998 and 1999, and that's this poster up here titled "The Survival of Staphylococcus aureus in Acidic Foods Through Inducible Acid Tolerance." Now this poster was presented at the 2001 Australian Food Microbiology Conference in Melbourne. And this is work I did at the University of Ballarat, now Federation University Australia in Ballarat. What this work is about is showing the inducible acid tolerance system of Staphylococcus aureus and how that particular acid tolerance response can be induced in organic acid. So these are the two graphs that I've got here, and what you can see is in the green is the Staphylococcus aureus cells that did not undergo any induction or habituation in a low pH system. So I grew those in neutral pH, Brain-Heart Infusion broth 7.1, and then the red bars are the Staphylococcus aureus cells that I grew in Brain-Heart Infusion broth that had been acidified with, in this particular case, lactic acid. So Brain-Heart Infusion broth acidified to pH 5.5 with lactic acid, and in each case both of these sets of cells were challenged at a lethal pH with hydrochloric acid. So that's Brain-Heart Infusion broth acidified with hydrochloric acid, and at various different pH values and different times, so 15 minutes or 90 minutes. And in each case, while the survival is low for both sets of data, you can see a substantial difference between the green bars and the red bars. So on average the green bars show about 3 to 5% survival after the Staphylococcus aureus cells were grown in a Brain-Heart Infusion broth of a neutral pH 7.1. However, when those cells were exposed to a sublethal pH of 5.5 created with lactic acid in Brain-Heart Infusion broth, they were able to induce an acid tolerant state which enabled them to survive better when they were challenged at a normally lethal pH of either 3 or 4, for example, and I've got 6 in there as a bit of a reference. But you can see that the percent survival is between 15 and around 23% compared to between 3 and 5%. So a substantial increase was seen. So from this you can see that Staphylococcus aureus is capable of inducing its acid tolerant state in organic acid. Now the other graph I've got up the top there is what might happen in food. So this particular system was a artificial system in Brain-Heart Infusion broth, true laboratory study, if you want to put it that way. Whereas the one up the graph up the top is a food study in salad dressing. So what I've done there is adapted the cells, so exposed the Staphylococcus aureus cells to either Brain-Heart Infusion broth at normal pH 7.1, neutral, close to neutral, or the Brain-Heart Infusion broth that had been acidified to either pH 4.5 or pH 5.5, in both instances with lactic acid. And then they were subsequently challenged for either 2.5 hours or 7.5 hours in a salad dressing, which has a pretty low pH. So this was to show whether the cells that had been exposed to a sublethal pH with organic acid, in this case lactic acid, were able to survive. Were able to have an increased survival in a food system, in this case salad dressing. And you can see that it is it's pretty significant, because the green bars there, after 2.5 hours, there was around 5% survival in salad dressing after the cells were exposed to normal neutral pH Brain-Heart Infusion broth. However, the cells that were exposed to Brain-Heart Infusion broth with pH of 4.5 acidified with lactic acid had a slightly increased survival, around 8% compared to 5. But if the cells were adapted in 5.5 pH, then you can see this particular bar here is around about 32%. So nearly a third of the cells, nearly a third of the Staphylococcus aureus cells survived in salad dressing after 2.5 hours if they were exposed to pH 5.5 acidified with lactic acid, compared to exposure to a neutral pH. And in the case of this result over here, the 7.5 hour challenge in lactic acid pretty much knocked out the cells that were the control cells, so the cells that were exposed to Brain-Heart Infusion broth at the neutral pH, as well as the cells that were exposed to pH 4.5 acidified with lactic acid, whereas the cells that were exposed to pH 5.5 had they didn't have a great deal of survival, but they were around 2 to 3%. So some did still survive compared to the others. So up until this point, what work had been published was that Staphylococcus aureus could induce an acid tolerant state in a inorganic acid system, so typically hydrochloric acid. But in my work here, I demonstrated that Staphylococcus aureus could in fact induce an acid tolerant state in organic acid, which is of particular relevance to the food manufacturing industry, since it's organic acids that are present in food systems. And if we reach over here, just to show, this is my honours thesis, which contains the full data, the full raw data, and all the information about this work and the other work that I did at the what is now Federation University Australia. So that's it from me, Dr. Philip Button from the Food Microbiology Academy. Thanks very much for listening, and I'll see you next time. Thank you.